Hello, everyone. How are you doing? We are live in Zurich, Switzerland. Thank you very much for joining another MaxWell webinar. So, what is our agenda for today? Okay, so first, my name is Marie. I will be your host for today's webinar. And for the introduction, I will introduce to you our company, MaxWell Biosystems, and our technology, microelectrode arrays. Next, we will have the main topic, which is a presentation by one of our application scientists, Dr. Szilard Sajgo. He will talk about label-free functional characterization of 3D organoids at single-cell resolution. This is a very hot topic right now, and we are very excited because we know that in neuroscience, developmental biology, organoids is one of the most important key important updates development that is going on right now in the scientific world. Okay, so just a note, this is the outline, and by the end of the presentation, we will have a question and answer part wherein we will look at the chat room. So anytime during the webinar, please feel free to type in your questions and we will answer them. If ever we are not able to answer all of your questions, please don't worry, we will send you an email and contact you directly. Okay, so we also have right now a brochure which is related to organoids. So most of the results that you will see here are in a brochure. We will have here the download link in the chat room as well. Cool. Okay, so let's begin. So we are MaxWell Biosystems. We are based in Switzerland, and our mission is to provide electrophysiology systems and solutions to advance scientific discovery and to accelerate the development of novel therapies for brain diseases. Our core expertise include microelectrode array platforms, both on the technical side, which is hardware and software for microelectrode arrays, and also on the application side. And our technology is basically a way to which allows us to characterize cells. So these are the two different ways to characterize cells nowadays. First is patch clamp, wherein you get the function of the cells. You have a micropipette that goes into the cell to be able to record these signals that you see here, which are mostly action potentials. On the other hand, we also have imaging that allows us to see where the cells are, how they grow, and how they connect with each other. However, it's quite difficult to get single-cell resolution activity from such imaging systems. So what we have is a microelectrode array, high-density microelectrode array that allows us to do both electrical imaging and also getting the electrophysiology. So we record action potentials from individual cells, and we're able to extract the propagation of such signals along the axonal arbor, which looks like this. So this is an action potential originating from the axonal initial segment and propagating along the axons, which allow us to basically extract the morphology of these cells using the technology, using the microelectrode array. And imagine that you can do this for hundreds to thousands of cells from your sample. All right. So just again, looking into what we measure. So it's electrophysiology. So intracellularly, we have the micropipette inside of the cell to be able to get these action potentials, which you see here as a positive signal. Extracellularly, we have these electrodes that detect the same signals from outside of the cell, which we see here as negative spikes. So each one of these is an action potential. And because we have many electrodes, not only can we have one signal per cell, but actually we have waveforms across the entire morphology of the cell, which looks like this around the axon, close to the soma, and also along the dendrites. So what are the key advantages of high-density microelectrode arrays? So other MEAs are lower resolution, wherein you're not able to see all of the cells. So with high resolution, you can see all of the cells, and you can identify the activity of each cell, whether it's coming from the green cell, the red cell, or the blue cell, as compared to having just one electrode recording from these cells. You're not easily able to identify if you're recording from 3 cells or from 1 cell or from more cells. Also, with many of these electrodes, you're always at the right spot where there are cells. And because we have small electrodes, we have access to cells' small signals. This is the reason why we're able to identify the propagation of the action potential along these axonal arbors. In this way, we are able to extract readouts across different scales at network level, which is basically like population spike times, also bursts, at cellular level, which is individual spike times, and also individual spike waveforms, and even at subcellular level, which is just the spatially resolved waveforms across an individual, across the morphology of an individual cell. So this is the main advantage of using high-density microelectrode arrays. So what are our products? We have MaxOne, which is our single-well microelectrode array, and MaxTwo, which is our multi-well system. So MaxOne has a recording unit, which is very small, similar to the size of your cell phone, which can go inside of the incubator if you're doing cell cultures. And it can also be used for brain slices, retina, and so on. This is our consumable, which is a single chip wherein you can put your samples into. So you just plug it in into the recording unit to do the recordings. And then for MaxTwo, we have our mainframe, which is also a built-in, which has also a built-in incubator. So you can have the same environment for the cells as it is inside of your main incubator. And for the well plates, we have 6 and 24 versions. So zooming in into the microelectrode array, in each of the well, we have a 2 by 4 millimeter square microelectrode array, which has 26,400 electrodes. And the distance between two electrodes center to center is 17.5 micrometers. So you can imagine that for one cell that looks like this in green, there are multiple electrodes that are able to detect the signals from that single cell. And very importantly, we have low noise readouts, which is why we're able to detect these small signals along the axons. And any of these 26,400 electrodes can be used for electrical stimulation. So you can choose them selectively and identify which ones are closest to the neurons that you would like to evoke action potentials into. Okay, and central to our systems is the software MaxLab Live, which is an all-in-one software that allows us to control the hardware and also do the recordings and also data analysis. So with MaxLab Live, you're able to visualize and acquire high-quality data, organize experiments, analyze the data, and also generate summaries or reports from very basic analysis. So what can we do with MaxLab Live? First, we can visualize the data that look like this. So electrodes view, each of the squares here correspond to an electrode. So you see the activity of all of the cells on the array. And we also have the raster plot view that allows you to see the population activity across 1,000 channels. And this is very cool. Another visualization, which is just the electrodes color map. So here you can see the activity of each of the cell. So each spot corresponds to a single cell, not a population of cells. And you see when there is a burst happening, which is really, really cool. Okay. Aside from visualizing data, we can also perform assays. And for today, we are showcasing neuronal assays. We can do activity scan, network recording, and axon tracking. So what are these assays? So for activity scan, the first question that we ask is, are the neurons active? So here what we do is we scan all of the 26,000 electrodes to be able to extract the whole sample activity map, which is either a firing rate or a firing or spike amplitude for each of the electrode. So here in the image, you can see a fluorescence microscopy image wherein these spots are where the cells are, which nicely correspond to the functional image that you can extract using MaxOne or MaxTwo. So here, each of the pixels would have a value, which is in this case firing rate, and the red spots correspond to the most active spots, and the light green spots correspond to the medium active spots. So once you know where the active cells are, you can then choose your top neurons, let's say 1,000 neurons, and then you can continue on with the next assay, which is the network connectivity assay. Here we ask, are the neurons interconnected? And we can answer this by first looking at the bursting features, how synchronous are the firing of these cells, and then we can also extract a functional network connectivity from each of these cells. So what are we seeing here? So the spiking activity for each of the channel looks like this. And whenever there's an action potential detected, there is a dot that is represented here in the raster plot. So each line corresponds to one channel that looks like this. And whenever you see these vertical lines, they correspond to a network burst. The summary of these network bursts look like this, which we call network activity. So when this is high, that means all of the cells or most of the cells are active at the same time. And when it is low, the activity is quite low for most of the cells. So the next assay is axon tracking assay, now really zooming into individual cells. And here we ask, are the axons functional? And here, we extract axonal arbors of single neurons and calculate also the axonal action potential propagation velocity. So we are able to extract the morphology of the axon. In this case, we can even follow the same cell, let's say from day 17 to day 30, and see that these axons are growing, the branches are growing as well, popping up, and also we can compute the velocity of the action potential propagating here. So this is an example of an action potential propagating across multiple electrodes. So the timing of these spikes would be the way for us to compute the velocity of the action potential propagation. Okay, so these are the things that we can do. And of course, we can do basic analysis also within the software and generate these results. So neuroscience applications, 2D cultures, such as from human iPSC-derived neurons or primary neurons, from 3D samples, such as brain slices, brain organoids, and neuronal spheroids, and also from the retina, wherein we can combine it as well with a light stimulation setup. So, and for today, we will focus on brain organoids. And the topic is label-free functional characterization of video organoids at single-cell resolution. So let me welcome Dr. Szilard Sajgo. Szilard?
Hello, Marie.
Hi.
Hello, everyone.
Thank you so much.
So today I will talk about label-free functional characterization of 3D organoids at single-cell resolution. And to begin with, you might ask the question, why study organoids? Why study human organoids, and what is their relevance? So one of the major challenges of neuroscience research when trying to develop new therapies, or even during performing basic research, is the reduced translatability. And this usually originates from the increased complexity of the human nervous system and the fact that most neurological disorders are cell type or circuit specific even. And currently the standard research pipelines include two general approaches. One of low throughput, which uses human or primate samples. With these methods, scientists can generate hugely significant, can generate data with hugely significant relevance due to the known physiological and morphological cell types. And then you have the other approach of high throughput by using animal models. These can provide experiments with increased reproducibility, but in many cases, reduced relevance. Just to mention the example of the mouse model, for instance, used to study age-related macular degeneration. And in this case, the only problem is that the mouse does not have a macula. So you can see the problem with these model organisms sometimes. And where do organoids fit in this picture? With their three-dimensional architecture, they can be used to more accurately model physiology and development of different organs. And in most cases, they allow for the development of multiple cell types. One can just note, for instance, the retina organoids pioneered by Dr. Sasai, where you can have all major cell types of the retina within the organoid. The other advantage of using organoids is that you can reproduce the exact genetic background of patients. Thus, you can significantly increase the clinical relevance of experiments on organoids, and they can be used in relatively high throughput. So how can you use organoids in combination with MEA technology? What can you actually read out? So your two major readouts by placing the organoids, of course, on the surface of the MEA are the local field potentials, which measure the aggregate synaptic currents, or the action potentials, basically, as Marie mentioned, the extracellular activity of the organoids. And one might ask, what aspects of biology can you study using organoids in combination with microelectrode arrays? So I will give you some examples where MEAs have been successfully used in combination with neuronal organoids, but not limited to. For instance, in drug screening, in analyzing disease mechanisms, in toxicity testing or toxicity screening, in regenerative medicine, development, and even infectious disease, hugely relevant nowadays. So as a first example is the paper of Durance et al., where authors use serum-free embryonic bodies. This is before you have your organoids, a step earlier, that mimic early cortical development and can present the basis for a screening platform that can lead to better understanding the etiology of certain neurological diseases and how they originate during development. And in this study, the authors aim to set up a screening platform with these organoid bodies. And as proof of concept, they do show that GABA antagonists like picrotoxin increase mean firing rate of organoids, NMDA or AMPA inhibitors, basically they decrease the mean firing rate of organoids. And they also showed that in these organoids, they were able to induce the chemical LTP with glycine that basically also increased the firing rate of these organoids. So this is just a very nice example of how new platforms are being developed in high throughput to use organoids in high throughput screening methods. Another paper by Shurenko and colleagues with the similar principles demonstrate the possibility of use of organoids for toxicity testing, where they test 91 compounds on organoids and MEA, including drugs, pesticides, flame retardants, food components, and so on. And they are able to show how different compounds act on the firing rate of these organoids. What is highly relevant is also regenerative medicine and the use of organoids. For instance, in this case, what the scientists and Hernandez et al. Have done is they have taken iPSC cells from patients, they have created retina organoids, they have transfected them with JAWS, which is a hyperpolarizing chloride channel, sensitive to red light, so basically a redshifted rhodopsin. And then they have dissociated these organoids. They have taken the photoreceptor cells, transplanted it in a blind mouse model, an RD1 mouse model, and they were able to show that this retina in this mouse model basically, again, is showing light response. Now, you can also use these organoids to highlight different aspects or study different aspects of development. For instance, authors in Trujillo and et al. Demonstrate the potential of cortical organoids and how they can advance our understanding of functional electrophysiology during different stages of development. And they also demonstrate that certain brain organoids very closely mimic fetal development. So this really opens up the window for studies that normally you could not do during fetal development, embryogenesis. Another interesting application of organoids and MEA is studying the human cytomegalovirus-induced microencephaly in organoids. Because this virus has high tropism towards human cells, it is not really possible to study it in animal models or mice, or quite difficult. And in this case, the scientists derived organoids. They infected it with the virus. They have placed the organoid on the MEA, and what they observe is that these organoids, once infected, they lose electrical activity. And they are also able to show that in case they use 1B2 neutralizing antibody that basically kills off the virus, then these organoids basically regain their activity, and they are able to show this with the MEA. Now, as mentioned by Marie, unlike in conventional low-resolution MEAs, our system allows you to record the organoid's electrical activity independent of placement. Basically, as you are always at the right electrode at the right place. And as evident in these pictures, you can see a beautiful organoid on the left image, and then you can see the firing rate and the amplitude. of these organoids in the heat maps on the right. So this is the automatic activity scan that we provide, and you can very easily assess your organoids electrical activity by. And as Marie mentioned, you also have a network assay. Then you can use to access the network bursting activity of the organoids. And for instance, in the previous slide, the organoids I've showed you, in case you look at the baseline network activity, what you can see here on the left, you see beautiful synchronized firing. But now if you use NMDA, you apply NMDA, you increase the mean firing rate and the spike rate of these organoids, but you lose network activity. And for instance, if you use disoclopin, which is MK801, you both decrease the network burst activity, also the mean firing rate, but interestingly, the remaining spikes, they do not change in amplitude. So with this easy assay, you can relatively quickly assess your drug effect on your organoids. Now, I will also present you a study that was performed by Ana Rita Gomes from the Stem Cell Engineering Research Group at IEB in Lisbon, in Portugal. And I would like to thank her for providing us with this beautiful data. And this study is on trying to elucidate disease mechanisms. And the disease that they are focusing is the Rett syndrome. Rett syndrome is a neurodegenerative disease, and it affects 1 in 1,000 live births. And it's mostly caused by MECP2 gene, a gene on the X chromosome. And what are the phenotypes? The phenotypic representation is a loss in learned motor and language skills after the age of 2, severe seizures, breathing abnormalities, cardiac defects, and dendritic morphology changes in the cerebral cortex and cerebellum. Scientists until now have been focused on animal models, but these have the disadvantage of not fully recapitulating the phenotypes. Also, this being an X-linked gene, it's relatively hard to study only on male animals. In two-dimensional iPSC culture models, again, they fail to recapitulate the structural complexity of this disease and the cells of the human cortex, whilst the previous studies on three-dimensional organoids, they failed to reproduce the axis organization of the different brain regions that is thought to be of significant relevance in this disease. Now, new protocols emerging. It is possible to generate forebrain organoids with dorsal and ventral axes in a sort of a modular system, where scientists develop these dorsal and ventral organoids or cerebral organoids independently, and they assemble them within one organoid and create these assembloids. And they can do this with forebrain, midbrain, and cerebellum samples. This allows them to study interregional interactions, like how neurons from the forebrain and the midbrain interact, whether they migrate from one area to the other, and so on. And they can study this in these assembloids in healthy and diseased conditions. And they can also aim to develop a drug screening platform using these organoids. And as you can see on the images, we do see a dorsal and a ventral organoids, ventral organoids being labeled by GFP in this case, via a virus infection, but scientists can also use specific ventral promoters like MKX2 to do this. And then you can see that after a day or so, or 2 days, these organoids fuse. Now, once you take these fused organoids and you place them on the MEA, you can very easily assess again their activity, their firing rate, the spike amplitude, but you can also very easily access their network activity. And as you can see here in the MECP2 mutant, the mean firing rate and the spike amplitude are significantly reduced in these male organoids, and this is related this relates to the absence of neuronal oscillation. So these neurons start to lose neuronal oscillation capabilities. Now, besides the fact that you can study these organoids and you can study this disease, what you can also do is use the MEA system for kind of protocol optimization. For instance, what scientists have done during the development of cerebellar organoids is that these protocols were not perfect. Neurons tended not to have synchronous firing. Purkinje cells had not suboptimal morphologies and suboptimal connectivity maps. And then scientists figured out that once they add serotonin, as you can see here also in the activity map and in the network activity map, that once you add serotonin to these organoids, you do regain synchronous activity. And this is in line with the mouse data that has shown that, for instance, 5-HT receptors, serotonin receptors, are really necessary for proper Purkinje cell morphological development and connectivity development. So in this way, you can use the MEA also to optimize your protocols. Now, you can also use the system due to the large number of electrodes to very easily track the activity of single neurons in brain organoids. And as you can see on the image on the left, you can very easily extract the extracellular action potential spatial maps, the distribution of this. And by using different spike sorting algorithms, you can again relatively easily extract the footprints of neurons. In case you have processes, you can show where these processes project to, and you can really analyze your data at a single cell resolution. Now, just to recapitulate, I hope I have demonstrated to you that how high-density MEA from MaxWell Biosystems can be at the center of your organoid physiology needs. And with this, I would like to thank you very much.
Thank you very much, Szilard. That's really, really cool. Again, thank you very much to both Hobson Biotechnology and also to Ana Rita Gomez for all of the results that we have shown here today. I will go through all of the questions. It's very exciting. I think it's really because our topic is very hot right now, so we are getting a lot of questions. But let me just kind of recap all of the things that one is able to do using microelectrode arrays, high-density microelectrode arrays, MaxOne and MaxTwo with organoids. So as Szilard mentioned, first, we can just first identify if the organoid is functional. Second, we can test drugs on these organoids. Third, we can characterize functional phenotypes of different types of organoids. And fourth, we can also optimize protocols in developing different organoids. So, okay, now let me go to the questions. Okay, cool. So the first one we have here, we have a great image of neurons laying on the electrodes. Can we electroporate the pads and create a different surface geometry for neurons to grow on? Maybe I can answer first, and Szilard, you can add to that. So as I mentioned earlier, any of the 26,400 electrodes can be used for electrical stimulation. Now, depending on the protocol that you use for electroporation, we can see if that fits with the ranges of either voltage or current that can be delivered through these electrodes. And we have 32 channels that can be used at the same time, 32 stimulation channels. So you can then use multiple electrodes to stimulate some area on the array at the same time. Yeah, so maybe to further discuss this, we can discuss later on. Szilard, do you have anything to add?
Oh, I think you covered it.
Okay, yeah, thanks. Next question. So this is really cool, and I'm also happy to hear about this. So 3D organoids are three-dimensional; they're not flat. So what are different ways to place them on the array to be able for us to record from them?
So there are multiple possibilities, of course, depending on what type of organoid I'm recording. And this will differ mostly in the size of the organoids. So you can either culture the organoids by using standard coatings and standard cell culture approaches, or you can also use the MEA to perform acute experiments. In case that you are performing acute experiments, the most important aspect is that once you place your organoid onto the surface of the MEA chip, it's recommended to use some highly absorbent pads to make sure that you remove the residual liquid between the organoid and the MEA, thus creating a perfect contact. And after a couple of seconds, you can add your media back, and you can record from your organoids up to 1 to 2 hours in an acute aspect. For other types of organoids, for instance, retina organoids or small organoids, you can also use these small tissue harps to keep them on the surface of the MEA, again, to uh, use it in acute preparation. Or again, you also have the possibility of culturing organoids on the surface.
Thank you, Szilard. Is it also possible to make organoid slices and place them on the array?
Yes, that is also possible. The only issue there is that you need to make sure that the way you slice your organoids, you don't distort too much the morphology, because the smaller the organoid is going to be harder to section them and to maintain integrity.
Yes. So for all the questions regarding protocols and also how to place the organoids on the array, please feel free to contact us, and we can find a solution based on the type of organoid that you have. Okay. Next. Oh, this is cool. So in one of the slides, we have the picture of the organoid, and also with the activity maps overlapped. How is it able? How are we able to take a picture of the organoid and align it in the software window?
This is relatively easy. It's as easy as using a smartphone. If you can take a close-up image, MaxWell Biosystems has an option to load this image and to automatically align with the electrodes, and then you can just superimpose the activity scan results with your image.
Yeah. So the way it's for the alignment is just basically clicking onto the four corners of the microelectrode array. That's it. Yeah. Of course, you can also use a microscope to take these pictures to be very precise. But if it's just the placement of the organoid, you can use any camera. Cool. Okay. Now, next question. So again, the sample is 3D. Don't you only see the neurons that are directly in contact with the MEA?
So because our MEA's electrodes are really sensitive, you do record signals up to 100 micrometers deep. But of course, if you want to go beyond 100 micrometers deep, you might need to section your organoid to really see what's in the middle of the organoid.
Yes, and it's just that, of course, the deeper the cells are, the smaller the amplitude of these signals are, and yeah. Okay, next question. Does the organoid move a lot during recordings?
Again, as I mentioned before, if you use these absorbent pads and you manage to absorb most of the liquid between the two surfaces, they should not move. Again, this is going to depend on the size of your organoids. For smaller sized organoids, you do need to put a coating and make sure it sinks and attaches. But if you do organoids that are 4, 5, 6 millimeters across, then this should not be an issue. Again, these need to be optimized for your individual needs and your individual organoid types.
Yep. Okay. Next question. This I would have to ask, but we will try to answer in both Szilard's presentation and my presentation. So here the question is, the synchronized bursting that you showed looks super periodic. Is it common with such cultures? Um, I think the one that Szilard showed is not really very periodic, right, Szilard?
Yes, it's not very periodic, but again, this will highly depend on your organoid and your maturation level of your organoid. And even in the same batches, you do have variation. You can have organoids from the same batch that show nice rhythmic activity and nice activity in general, and you can have ones that show reduced activity. So, I mean, these protocols are not perfect yet.
Mhm. And for the data that I have shown in my slides, these are actually not from organoids, but from 2D cell cultures. And the periodic bursting that you have seen is actually from iPS cells, a specific type of iPSC-derived neurons. Yeah. Okay, next. Oh, cool. Is it possible to point out the technical points of the brain organoid on your MEA? Meaning, are the organoids cultured on the MEA the entire time, or are they formed into a sphere and then transferred?
So in the presented cases, they are formed into a sphere and then transferred on the MEA and cultured for additional 3 to 4 weeks on the surface of the MEA.
Okay. And as a follow-up question, are these recordings in the media?
Yes, of course, the organoids are in the media, or again, depending on application, you can also have brain freeze media or your media of choice.
Mhm. And next, is there no need for a special ECM or matrix or gel to get them to interact with the MEA with enough sensitivity in case of acute?
In case of acute, no.
Okay, but for culture,
For
yes.
culture, yes.
Mhm. Awesome. Okay. How long can you record continuously?
Again, if you're doing it acutely in an incubator, I would say several hours, maybe even more. If you're actually culturing the organoid, I would say for days, as long as the organoid stays on the surface of the MEA. You do need to keep in mind the file size generated if you do 5, 6, 7 hours of recording.
Yep. Okay. And that's basic network recording, right?
Yeah.
Yes. Next, we have so many questions in our popular topic. So, can you also monitor the cell differentiation using this system?
In theory, yes. If you culture the organ on the surface of the MEA, you can do recordings. You can assess at what time activity starts to pick up, and then, of course, if you are able to maintain the organ in the MEA, you can record until a point where you see synchronous activity.
Okay. Still more questions to come. How long can you keep the organoids alive on the MEA?
So far, I would say for months.
Yep, yep. I guess again it depends on the protocol and the type of organoid, right?
Yes,
So yeah.
exactly. And the stage of differentiation is.
Yes. Cool. Next, how do you add the molecules?
For drug testing,
I guess so.
you just very precisely dilute it into the media that's in the chamber of the MEA. And of course, you can also have a drug delivery system similar to what you would have in case of a patch pipette.
Mhm.
You just need to have a proper doser, and that's how you do it.
Yep. Okay. Is there a best way to seed a spheroid into a specific location on the array?
I would say practice. The issue here is that once you place the organoid on the MEA, and you try to align, if it sinks and if the chip is coated, then I would not move it again. If it's acute, you can potentially put media again and let it float and try to orient it again. What I recommend is just use a small tiny brush or sterile capillary filler tips to try to orient it. And then once you're happy, you can remove the liquid and help settle the organoid.
Yep.
But it's mostly practice.
Yes. Yep. Next, how do you know the shape of single neurons in an organoid?
You perform spike sorting. You take the data and you use spike sorting algorithms, for instance, so spiking circuits or kilo sort or Plexon or whatever you prefer, and then you extract the waveforms. Each neuron has quite relatively different waveform, and these algorithms they cluster them, and then you can, of course, plot back the activity map, and you can see that these waveforms are coming from which electrode, and then you're more confident that that's one cell.
Mhm.
And because you have so many electrodes, then you can, when you do these clustering algorithms, your clusters are much easier to clean and to separate. In case you
Nice.
have a large electrode and you have 20 cells on one large electrode, then this clustering becomes more and more difficult.
Yes. And Szilard is also our expert in spike sorting. So if you have more questions about that, please feel free to ask Szilard or send an email to info@mxwbio.com. Okay, so I think we will close the session quite soon. Let me just go to the last question. There are still some, but we will answer them directly by email. So for our last question, we have, what is the advantage of acute recordings versus long culture?
Acute recordings, okay. So if you culture your organoid on a planar surface, then these organoids have the tendency of losing their structure, their basal structure. Now, if you record acutely, you can place the organoid on your chip, you can do your recording, apply your drug, and then you can again remove the organoid once you're done, put it back into your culture chamber, culture it for a couple of more days or weeks in optimal conditions, in most of the cases on a shaker or a rotator, and then you can place it back. So, and this way the organoid maintains its shape and it's much easier to do.
Yes, it's just that for cell cultures, I guess you're able to see also kind of the same spots in
Yes,
time,
yes.
while for acute, you're not able to do
Again,
that.
it differs from what do you want to see. If do you want to see progression of network connectivity, or do you want to see the progression of these regional neurons in this organ? So
Okay. Thank you very much, everyone, for all of your questions and for your attention. Just a few things here. So next week, we will be in two virtual conferences. We have SPS, Safety Pharmacology Society, so we will have a booth there, and also Discovery on Target. We will have both a booth and also Szilard will have a presentation there as well next week. And for any questions that you have, please feel free to contact us at info@mxwbio.com, schedule a call directly with any of us through this link in Calendly, or visit our website. And once again, we have a brochure with all the results that you have seen here, so please download that in the link that we have shown here in the chat, or also from our website. You can just look at publications and you can see it there. And finally, for our next topic for the webinar, on October 15th, we will have a very exciting webinar again, which is based on the latest results with human iPSC-derived neurons. And we will send you more information about this, so please stay tuned. And we are very excited for everyone attending this conference, this webinar. And yeah, see you again for our next event. Thank you very much.
thank you.
Bye.
Bye bye.